If we look at now another study that also looked at this Mon 10 that was also published last year, this is again over 10 years after these crops were approved. Nobody had looked at this before. This was also a well-designed study because this Mon 10 and the near isoline were grown simultaneously in neighboring fields in Ladriano, Italy. That controls for environmental effects. And what they did is they did feeding studies with mice that were, they looked at weaning mice and older mice because they wanted to see if there was any stress happening, and they looked at, they were fed a diet containing Mon 10 or its parental isoline for 30 or 90 days. And there were all these alterations in T and beta cells and all these other subpopulations of cells. They said there was an increase in serum, a number of the intraleukins, and as they concluded, these results suggest the importance of the gut and peripheral immune response to GM crop ingestion, as well as the age of the consumer and the GMO safety evaluation. Again, unintended effects from a simple one-gene insertion that was not adequately studied. So this assumption that even the parental lines have been looked at carefully turns out not to be true. If we then now switch over, this as part of the ad hoc task force on foods derived from modern biotechnology, the consultation, one of the major conclusions was that all foods must be assessed for allergenic potential. They also recommended that they use this decision tree that was developed for determining allergenicity. Here's the decision tree. The only thing I'm going to talk about, because I don't have time, is the first screen is you look for sequence homology. And basically, genes are producing proteins. A protein is a string of amino acids. So one of the first things you should do is you look and you say, well, I'll look at the protein that I'm inserting into this plant. Does it have sequence similarity to known allergens? The following year, there was a scientist from the Netherlands which looked at these transgenic proteins. They looked at these crops. They looked at a number of transgenic proteins, and they looked at crops with known allergens. They're looking for these identical sequences of at least 6 amino acids. Anything that came up positive, since you might have false positives, they ran through further screens. They either looked at an epitope database. What an epitope is if somebody has an allergic reaction to a protein, you're not responding to the whole protein. You're only responding to little tiny pieces on the surface. They can be as few as 4 or 5 amino acids can be what actually triggers the immune system response. And what they looked at is they looked at 33 transgenic proteins. 22 out of the 33 had significant had identities of at least 6 contiguous amino acids. These are all the traits. You can look and see all the cry proteins. Cry 1AB, 1AC, 1FA, 2AB. So a number of them are showing up. There were 83 identities with allergens. When they then did the 2 further screens, 4 proteins popped out. There's one of the cry proteins. Cry 1AC is at the top. And those 2 peptides were identical with cedar pollen allergens, and so they said that those 2 sequences, they could potentially cross-link and cause an allergenic response. That just means it should be looked at further in further studies. And Dr. Stephen Jandel, who's head of the Biotechnology Studies branch at the U.S. Food and Drug Administration, the research side, he also did work looking at these things and noted that the similarity between Cry 1AB and vitellogenin, that's sufficient enough to warrant additional evaluation, because in there was a homology sequence of, I believe, 9 amino acids. And we can also look with these Cry proteins. There has been some evidence of allergenicity. This is work done about 10 years ago, looking at farm workers in onion fields in Ohio that were exposed to BT sprays. And they found 2 out of the 123 farm workers had actually IgE antibodies to the delta endotoxins. And they concluded that now we have the reagents and we can actually look at safety assessments for these various crops. Ever since this work, which this study was actually co-funded by the U.S. Environmental Protection Agency and Health Canada, since this time, Dr. Bernstein has not been able to get money anywhere to do the studies. In fact, he pointed out that workers in agricultural and food preparation facilities who are inhaling dust from these engineered plants, they could be exposed, and he said that the potential for occupational and consumer risks needs to be assessed. I'm going to have to go through this data very quickly. This is work done in India where they looked at 5 villages, talked to people with symptoms that had exposure to Bt cotton. And again, this has a cry protein. These cry proteins are also found in corn and other compounds. And all the folks had skin symptoms, itching, redness, eruptions. They occurred on the hands, face, hands, and feet. Half of them had symptoms of the eyes, itching, redness, swelling. 40% had upper respiratory tract symptoms, and actually symptoms of the skin, the eyes, and the upper respiratory tract. Those are classic allergy symptoms. 90% of them, 45% had moderate symptoms, and another 45% there were severe symptoms. They were overwhelmingly on the exposed parts of the body, which you would expect. 80% of them had exposure in cotton fields, and 4 of them were being exposed at homes, because in these parts of India, when they harvest the cotton, they'll bring the bales often inside their houses to dry. And as I said, 3/4 of them were directly involved in picking cotton. And probably the most damning thing is the fact that their symptoms increased in severity when they continued to work in the fields, and when they stayed away from the fields, the symptoms declined. So that suggests that it is due to this exposure. And the symptoms only started within the last 2 years, which is exactly when Bt cotton was introduced. They also went to a ginning factory, and the owner noticed that most of the farmers were having skin-related problems. Detailed interviews with 6 workers found they all had itching problems, 2 were having eruptions. The workers had been at this ginning factory for 2 to 7 years, and they only started to see the symptoms in the previous year, which is, again, consistent with the introduction of Bt cotton. That suggests that there's something that should be looked at further. And then finally, I want to go through this quickly. The Environmental Protection Agency's Scientific Advisory Panel had a meeting this past February on data required to register plant-incorporated protectants. These would be pesticidal plants, the so-called Bt plants. One of the questions they asked the Scientific Advisory Panel is they developed a draft test guideline to determine the potential for synergism between combination PIP products. Like, if you put more than 2 cry proteins together, could they interact and actually have different sensitivities just to the target insects? And so they said, they asked the experts to comment on this methodology to support the analysis, whether they should do this or not. And as they said, this is from the minutes of the meeting. It says, in general, the panel believed that studies showed that synergism is not easily predicted, and therefore testing for synergistic effects when 2 or more PIPs are combined is warranted. And furthermore, in August of last year, they actually held some stakeholder meetings. I went to one of them, and they were proposing 4 different things, and this is one of them, that they are going to propose by next summer, that this will be required, and then it'll take about another 1.5 years to finalize that. But here you have the US EPA is recognizing there, yes, indeed, could be synergistic effects. They need to be looked at. And what's interesting is Monsanto and Dow were at this meeting. They knew that the EPA is going to move forward with this, that it will be required. So why haven't they done it with the SmartStax? So this really shows that there are questions about these crops that do need to be looked at, and that Codex does actually permit countries to require full safety assessments. So Canada can be vulnerable to any country that passes strong regulations. They can block any of these stacked products coming from either Canada or the US and not have to worry about a challenge at WTO, and I will end there. Thank you.